Hello everyone, my name is Ganesh Gautam, and first of all, I would like to thank our external examiner, sir, our campus chief, Mr. Hari Prasad Khanal, sir, my supervisor, Mr. Bisal Thaguna, sir, and all of my friends. My topic my thesis topic is utilization and production of wine from coffee pulp byproduct and coffee seed with their physiochemical and sensory analysis. So here are my table of contents. First of all, we will talk about introduction, then statement of problem, objectives of the study, significance of the study, literature review, materials and methods, sample formulation, preparation of wine, results and for discussion, and at last we will discuss about conclusions and recommendations. So let's talk about coffee. Coffee is one of the most important agricultural commodity in the world. 3.5 billion cups of coffee is consumed daily worldwide. Over 16 billion pounds of beans produced annually in 70 plus countries. Traditionally, coffee was grown under tree canopies, but after 1970s, modern sun cultivation practice is used, which have high yield but reduces biodiversity and requires fertilizers. Coffee grows best in equatorial zone called the bean belt, located between latitude 25 degrees north and 30 degrees south. There are mainly two types of coffee. First one is coffee arabica. It requires higher altitude and rich soil. And second one is robusta. It grows in lower altitude with warmer temperature. So coffee processing generates significant solid byproducts. Example: pulp, husk, mucilage, and byproducts can be utilized for alcoholic beverage production and cascara, coffee cherry tea from pulp. So, statement of problem. During the coffee production, large amount of byproducts, mainly coffee cherry husk, coffee pulp, mucilage, and processed water, are produced, which will affect the environment if those byproducts are not controlled or if not turned into some useful products. Fermentation, a natural way of mucilage removal, can produce large number of byproducts in the environment that can directly and indirectly affect the environment. It seems tiny, but on a large scale, the amount of byproducts also increases, so this is very critical. According to recent study, caffeine and tannins present in coffee are toxic to various aquatic organisms, while chlorogenic acids have negative effects on seed germination and plant growth. The processed water used to separate mucilage from bean in wet milling factories have a high BOD, which threatens water sources and aquatic organisms. Due to high contents of organic acids, caffeine, and tannins, the coffee pulp and processed water poses a serious environmental problems where the pulp is discarded in a landfill. Rotting pulp may lead to unpleasant smell. On the other hand, coffee pulp, a major byproduct, can be repurposed to make wine instead of being discarded. There are very few works on research of the coffee pulp byproduct utilization. Also, there are very few works on research of the coffee pulp byproduct utilization in Nepal. Objectives of the study. General objective. The general objective of the study is utilization and production of coffee pulp from coffee wine from coffee pulp, which is a byproduct, and coffee seed with mucilage, their physiochemical and sensory analysis. Specific objectives: to carry out physiochemical analysis of coffee pulp and mucilage, to prepare wine from coffee pulp and seed with mucilage using winery yeast, to study the TSS, acidity, alcohol percentage during primary fermentation, to carry out physiochemical properties of prepared wine sample, to carry out sensory evaluation and compare with market red grape wine. Significance of the study. Focus on utilize. This study mainly focuses on utilization of coffee cherry byproducts. It provides economic benefits to coffee farmers and processors. Coffee pulp can serve as a valuable, cost-effective, and eco-friendly raw material for the beverage industry. Instead of disposing coffee pulp as waste, it will be reused for beverage preparation, helps to minimize environmental problems caused by coffee byproducts. Research data will be useful for other researchers, scientists, scholars in recent related fields. Literature review. Coffee trees produce coffee cherries, which turns into bright red color when ripened. Cherries grow in cluster along the branches. There are layers of coffee cherry. First, exocarp, thick and bitter outer skin. Mesocarp, or called pulp, sweet and grape-like texture. Endocarp, parchment-like shell covering the beans. Silver skin, thin membrane around two bluish green beans. Endosperm, coffee shell. There are mainly three processing methods of coffee cherry: dry or natural method, semi-dry method, wet method. In dry or natural method, we harvest the coffee and directly send it to the drying and hulling. And in semi-dry processing method, we harvest the coffee beans, pre-sort, clean and clean the sour coffee cherries. Then we do pulping and mechanical mucilage removal. Then either we directly send to washing and drying and hulling, or we do a minor step called finish fermentation, then washing and drying and hulling. And in wet method of processing, in wet processing method, we use in wet processing method we do harvesting, pre-sorting, cleaning, floating, pulping, and instead of mechanical mucilage removal, we do a long fermentation, then washing and drying and hulling. So these are the products obtained after each steps, and these are the byproducts obtained after these steps. So literature, so chemical composition of coffee pulp. So according to Franca et al. 2009 and Murthy and Madhva Naidu 2010, this is the chemical composition of coffee pulp. Here we can see moisture content is 76.7%, protein 11.5%, fat 2.0%, ash content 8.9%, crude fiber 3.4%, total sugar 14.4%, caffeine 1.5%, tannin 3.0%. So materials and methods. First of all, raw material collection. Fully ripe coffee cherry sugar were purchased from Christie, Pokhara and yeast was provided from Pokhara Vigyan Tatha Praviti campus. And chemicals required: AR grade chemicals were obtained from Pokhara Vigyan Tatha Praviti campus laboratory. These are the chemicals used during the analysis: carbon tetrachloride, potassium permanganate, methanol, ethanol, sodium bisulfite, gallic acid, iodine, oxalic acid, sulfuric acid, sodium thiosulfate, caffeine standard, chromotropic acid, hydrochloric acid. Equipment required. Equipment were obtained from Pokhara Vigyan Tatha Praviti campus laboratory. These are some of the equipments: UV visible single beam spectrophotometer model 291, weighing balance, oven, incubator, gel digestion set, conical flask, pH meter, separating funnel, test tube, picnometer, bottles, water bath, intake serological water bath, and thermometer, refractometer, fermentation, glass jar, and cork. Preparation of coffee pulp wine. So first of all, we take fully ripe coffee cherry, then we do pulping, mechanical pulping. Here initial TSS is maintained at 16-degree Brix. Here initial TSS of the coffee cherry. Here initial TSS was 16-degree Brix. Then pulp is mixed with water at a ratio of 2:1. Brix is maintained at 18 to 20 degrees Brix. Prepared must is pasteurized at 65 degrees Celsius for 15 to 20 minutes. Addition of yeast: 0.25 gram of yeast per liter of must. Fermentation until TSS reaches 8 to 10 degree Brix. First racking for 10 days. First racking is done for 10 days. Pasteurized at 65 degrees Celsius for 15 to 20 minutes. Secondary racking, bottling coffee pulp wine. So this is the whole preparation of coffee pulp wine. Sample formulation. Here for control sample, I took 2 kilograms of black grape and I add water 2 liters, yeast 0.25 gram per liter of must, and sugar as per required. And for control sample A, that is coffee pulp wine, I took 2 kilograms of coffee pulp, 2 liters of water, and 0.25 gram yeast per liter of must and sugar as per required. And for the sample preparation of B, that is uh, coffee pulp plus seed plus mucilage wine, coffee pulp with mucilage and seed wine. Here I took coffee pulp 1 kilograms and seed with mucilage 1 kilogram and water 2 liters, yeast 0.25 gram per liter and sugar as per required. So these are some of the these are physiochemical analysis. Moisture content was done using by hot air oven AOSC 2005. pH by using digital pH meter. Total acidity by titrometric method FSSAI 2019. And TSS by hand refractometer. Alcohol content by method of AOSC 2005. Esters by titrometric method FSSAI 2019. Aldehyde titrometric method FSSAI 2019. And higher alcohol using by titrometric method FSSAI 2019. Methanol by colorimetric method AOSC 2005. Reducing sugar by the method of AOSC 2005. Crude protein by the method of AOSC 2005. S content by the method of AOSC 2005. Caffeine content by the method of spectrophotometer Hamsey et al. 2017. Tannin content by using the method of spectrophotometer Hamsey et al. 2017. Sensory analysis by following 9 points hedonic scale by submitter and panelists of teachers and students of Pokhara Tata Vigyan Pragati campus. So data analysis. Sensory evaluation was done by using 11 semi-trained panelists with 9-point hedonic ratings. Sensory score data was statistically analyzed through one-way ANOVA using SPSS version 20.0. Data analysis was done using IBM SPSS and Microsoft Office Excel 2019. So result and discussion. Proximate analysis of substrate. Here moisture content of coffee pulp is 79.17 plus minus 2.89. Black grape 83.60 plus minus 2.55. Mucilage 84.2%. Acidity percentage of coffee pulp is 1.5% plus minus 0.02%. And black grape 0.47 plus minus 0.01%. And mucilage 1.7%. Ash content 2.66 plus minus 0.09%. Yeah. And then sensory analysis. So for color, as you can see, sample B, that is coffee pulp wine, is winning with a 7.6 rating. And for flavor, also coffee pulp wine is winning with 7.4. So based on all these parameters, we can say that overall acceptance of sample B, that is coffee pulp wine, is highest because with 7.5 ratings. Physiochemical analysis of prepared samples. So alcohol content of control sample is 9.03%. Alcohol content of coffee pulp wine is 8.6 plus minus 0.6%. And alcohol content of coffee pulp with seed wine is 9.2 plus minus 0.2%. TSS of control sample is 9 degree Brix. Coffee pulp wine is 10 degree Brix, and coffee pulp with seed wine is 9.0 degree Brix. pH of control sample is 3.01, and coffee pulp wine is 3.27, and coffee pulp with seed wine is 3.16. So this is the fermentation kinetics. Fermentation kinetics. fermentation kinetics with respect to PSS. So this is the fermentation kinetics with respect to TSS. As here you can see on day 0 the TSS of each sample is around 20 degree Brix, and with the days passing, after 18 days the degree Brix of a sample, that is coffee pulp wine, is the highest and other two are similar. And this is the fermentation kinetics with respect to pH. Here we maintained our pH in around 6 in day 0, and after some days this is how the pH ranges. And why is let's see.